All right, where we left off at self-assembly and self-organization. Terms describing the autonomy and the programmed nature of building blocks in supramolecular chemistry include self-assembly and self-organization, which are comparable to Egyptian claims that the deceased is self-created, a fondling without knowledge of parents, and an unblemished birth. For he was not conceived, but simply the molecular sunbark exhibits its self-assembly and self-organization. Then, in the Amduat, the self-assembled sunbark is dragged by the 4 gods, then 7 gods and 7 goddesses, then 8 gods, and finally 12 gods and 12 goddesses drag the bark through the bowels of the serpent Anakteru. This may depict further formation and birth of molecules through covalent or covalent bonds when 2 atoms are able to share electrons, or through electrovalent bonds, which are held together between positively charged cations and negatively charged anions. As an example, if the sun god represents hydrogen and the 4 gods dragging the ball signify the valence electrons of carbon, then we have a covalent or covalent bond of hydrogen with carbon. If 7 goddesses representing nitrogen anions and 7 gods representing nitrogen cations then unite and drag the hydrogen-carbon bond, this may indicate a hydrogen-bonded molecular network formed with cationic and anionic building blocks. This thesis is called to supermolecular chemists to analyze the obvious chemistry in the Amduat, for it seems that the text may indicate some derivatives of HCN or hydrogen cyanide. J. Oro and A. Lascano Arojo reviewed the role of HCN and its derivatives in prebiotic evolution, finding the radioastronomical observations show HCN is a very abundant cosmic molecule that is the periodic precursor of molecules such as amino acids and purines and adenine and guanine. If this is the case, then the 7 gods and the 7 goddesses may represent the nitrogenous base of the purines, guanines, which always pairs with cytosine by 3 hydrogen bonds, or adenine, which always pairs with thymine by 2 hydrogen bonds. The catalytic process. Along with the deceased sun god's potential for self-assembly, his journey in the Amduat can be easily restated in the chemical terms of the catalytic process, which involves 3 steps: binding of the substrate, followed by its chemical transformation into the product, and finally the release of the product to regenerate the active catalyst. This is mirrored by the deceased sun god's binding with Osiris, the instruments of weaving indicating protein conformation, and when the transformation is complete, the release of the solar product comprised of Ra's loyal servants. In the same way, the least corrupt of all the funerary texts supports the idea of a product for the deceased king is a bright star bringing to Ra distant products on a daily basis. Similarly, the deceased makes his way in the valley as if he were a chemical reaction. He takes a minimum potential energy path up the floor of the reactant valley and down the floor of the product valley. Hence, the Amduat expresses an Egyptian awareness of the 21st century knowledge, such as molecular recognition, self-assembly, self-organization, and the catalytic process, which enable the pharaoh to identify quantum molecular structures using metaphoric language similar to that of our new chemists. Exploring requisite details, the sunbark as a gene vector. Exploring requisite details of the idea of the sun god as a lambda vector, we find that the lambda sun god and his supermolecular crew is a fusion that easily breaches the cell wall of the Earth at the gap of the duat. Bacterial viruses have this capability, along with the hierarchy of structural levels from molecular to the supramolecular, as do cells and tissue fibers. Since references such as Osiris's activity of eye and the eye of Horus suggest DNA activity, it is possible, as our motto posits, that the sun god and his crew represent a molecule capable of transporting DNA into a cell. A molecule of this nature is a gene vector, and modern geneticists favor the use of modified viruses which breach cell walls easily. So we could further describe the composite crew on the sunbalk as sunbalk equals lambda gene vector / supermolecule. If the supermolecular deceased sun god and the crew represent a lambda gene vector, then we must now identify the Egyptian meta symbol that signifies the transport of DNA. That is, the transcription, replication, and translation of DNA. We have posited that the union of the deceased and the sun god at the northern polar cusp parallels the insertion of the exogenous DNA fragment into the incoming lambda genome that will dock into the Earth cell. The deceased king clearly states, "I am merged in the sunshine god." The central 15 BP regions of the lambda genome between positions 20 and 35 is a non-essential region that controls the lysogenic phage properties. The lytic infection cycle can still function if this area is deleted or if exogenous DNA is inserted into it. For example, the insertion vector lambda GT10 has a single recognition site for restriction enzymes, allowing DNA fragments to be inserted into the genome. This recognition site is called EcoRA and lies within the CI gene or the lambda repressor. The CI gene is inactivated by cloning a fragment into the site. In the insertion vector lambda GT10, when DNA is inserted into the recognition site EcoRA, it generates left and right arms of 3.27 and 10.6 kb, which can accept a DNA insert up to 7.6 kb in length. Because the nature of the recombinant vector is to carry DNA fragments, geneticists use lambda vector for cloning experiments. In fact, modern geneticists have named insertion vectors such as lambda insertion vector Charon or Charon 16a, after the ferryman of Greek mythology who transported the spirits of the dead across the river Styx. The sun god is also the ferryman carrying the deceased through the duat along the host chromosome in a fashion similar to that of the cloning vector. So to learn more about the ferrying vectors, it now seems reasonable to compare the deceased sun god's entry into the duat with lambda events related to absorption, which involves the phage particle binding to receptors on the bacterial surface and injecting DNA. Absorption and injection into the duat. Recall from part 1 that FTEs, also known as bubbles, CDCs, or magnetic islands, are produced by tearing mold reconnection process driven by strong magnetopause currents and fluctuations of alpha waves. The FTE forms when plasma is advected towards the spiratrix of the inflow and injected into the region inside the spiratrix by the outflow jets. The result of this is the filling of the region inside the separatrix, with the plasma forming the FTE. Likewise, researchers Robert Sheldon defined a CDC as a magnetic bottle or force-free structure whereby the local plasma energy dominates over the magnetic energy, forming a bubble constrained by compressed magnetic field lines and increasing field strength along the field line. The origin of a CDC or magnetic bottle can be from an explosive heating event for a sudden injection of cold plasma. The sudden injection of cold plasma is the preferred option because of the presence of the magnetosheath plasma in the core. Similarly, when phage lambda absorbs into the bacteria cell, its tail fiber attaches to the lamb protein on the E. coli outer membrane. DNA is then thought to be injected down the hollow core of the tail into the bacterium. So if the staircase cuff signals the onset of a FTE, then its magnetospheric event may mirror the Egyptian idea of the deceased engaging the great stairway and becoming a recombinant with the lambda sun god and his supramolecular crew on the bulk. Further, the formation of the FTEs by the advection of the plasma towards the spiratrix may mirror injection of recombinant lambda DNA into the E. coli cell as the movement of the deceased god and the crew into the gap in the mountains, that is, through the cell wall. Just as the preferred origin of the CDC or magnetic bottle is a sudden injection of cold plasma, we find that the deceased king receives a jar magnetic bottle of Ra's cold water when he meets him at the polar region. In part 1, we analyzed a quotation from the Tibetan Recension about the leading moored one to the secret places and dragging and why, which seems a likely description of phage lambda attached and penetrating the whole cell. The text states that the dead king's boat is at the mooring rope and that he will not be boatless, and that he is stretched out straight and makes ready the ferry boat, which is moored in the dockyard of the gods. Ra also causes the gods to follow when the bark travels, it being dragged straight on. These parallels suggest that the journey into the duat begins at the site of impulsive magnetic reconnection at the cusp, which is similar to the injection of recombinant DNA into the E. coli cell at the polar lamb receptor site, a dockyard for phage lambda.